Good afternoon. It gives me huge pleasure to introduce James Finns. He did his PhD in Malcolm McConville's lab. He then came to my lab and was tremendously productive and beautiful cell paper in 2007, really driven by James' intelligence and hard work. And then he's gone on and been equally successful in Jörg Chopp's lab, and this is pretty much the story you're going to hear today. And I can personally say that I'm really looking forward to this talk because they've told me that there was some new data generated last night, and I haven't seen it yet. All right.
Thanks. Okay. As John said, this story that I'll talk about today began in Jörg Chopp's lab, who tragically passed away at the beginning of last year. And I was fortunate enough to be able to come back to Davo's division and work with Davo and John in finishing off the story. And most of what I'll present today has recently been published, but as John alluded to, there is some new data and some preliminary data in this story that we're following up on. And so this is I'll be talking about the role of death signaling in inflammation, and it's not your traditional idea of when you think about necrosis inducing inflammation. What I'll hope to present evidence today is to suggest that there's actually death signaling machinery that can directly influence inflammation by resulting in the direct activation of interleukin-1. So I'll just go through the well-accepted cell death pathways that have been studied for many years. So there's your intrinsic cell death pathway, which is mediated through back-to-back permeabilization of the mitochondria. And this results in the activation of the initiated caspase, caspase 9, and that leads to apoptotic or caspase-dependent cell death. And there's also the alternate cell death pathway mediated through death receptor ligands such as TNF, FAS, and TRAIL. And when these bind to their cognate death receptors, it initiates complex formation resulting in caspase 8 activation and again downstream caspase-dependent apoptotic cell death. Now these initiated caspases and effector caspases such as caspase 3 can be directly inhibited by the inhibitor of apoptosis protein, which is termed XIAP, and this can prevent cell death when this protein is expressed. And the cell's got a built-in mechanism to antagonize XIAP. So when the mitochondria membrane's compromised, the mammalian IAP antagonist, Smac, or also known as diabolo, is released from the mitochondria. And this binds to XIAP directly to antagonize XIAP function. And this will allow the activation of effector caspases and promote apoptosis. So based on these findings that Smac diabolo can antagonize XIAP function to promote caspase activation, a number of pharmaceutical companies have developed Smac mimetic peptidomimetics. These compounds bind to the baculoviral IAP repeat grooves within XIAP to antagonize XIAP function. So there's been quite a lot of interest generated in these compounds, and a number of pharmaceutical companies listed here have developed their own Smac mimetic compounds, and these have been in phase 1 clinical trials for a while now, and some of them, such as the one from Tetralogic Pharmaceuticals, which is listed here and which is who we work with, they're now taking their compounds into phase 2 clinical trials. And so this is just some representative data of what helped them get into the clinical trials. So this is a human cancer xenograft mouse model, and you can see here at low doses of these Smac mimetic compounds, they can prevent tumour growth or tumour formation in these mouse human xenograft models. So of course, these peptidomimetics were largely designed to antagonise XIAP function. But there are a number of IAP proteins within mammals, and two of the most commonly researched ones are the cellular inhibitor of apoptosis proteins, CIP1 and CIP2. Now these have a fairly conserved domain structure when compared to XIAP. They contain these BIR motifs, as well as a card domain and a C-terminal ring E3 ligase domain. So these BIRs mediate protein-protein interactions, and the BIR of CIP1 and 2 can regulate binding to TNF receptor-associated factors such as TREF2. As I mentioned, Smac mimetics or Smac diallo itself targets the BIR2 and 3 groups, and this E3 ligase activity of both XIP and CIP1 and 2 is critical for their function in regulating cellular processes. So what we discovered, and a number of other groups discovered with these Smac mimetics initially back in 2007, was that they don't actually just target XIP to induce cell death, and actually their targeting of cellular IAPs 1 and 2 is actually key for their induction of tumour cell death. And they seem to do this in two ways. They promote death receptor killing through the apoptotic pathway or caspase-8 dependent apoptosis by sensitizing cells to TNF receptor killing. And they also promote this NF-kappa B activation to promote autocrine TNF production. So within this TNF receptor associated complex that I've got here, TNFR1, CIPs bind to TNFR1 through their association with TREF2. And these CIPs mediate ubiquitination of this receptor interacting protein 1 kinase to ubiquitinate RIP1. And now this normally promotes NF-kappa B activation, and this NF-kappa B activation can induce genes that will block cell death. And what we showed, and others showed, that when you remove CIPs, you prevent the ubiquitination of RIP1 kinase. And this promotes the formation of what's being called complex 2, or the caspase-8 containing complex containing RIP1 and FAD, and this will initiate apoptosis. So recent work sort of took these studies further, and this was published in Pascal Myers and Martin Leverkuse's lab towards the end of last year. And they showed in some cancer cell lines that removing IAPs, and they had to remove XIAP as well as CIP1 and 2 for the maximal formation of this, what they call this ripoptosome complex. And they demonstrated when you remove IAPs with, say, Smac mimetic compounds, you get the spontaneous formation of a caspase-8 FAD RIP kinase and FLIC containing complex. And then this complex is poised to induce cell death. So in the presence of caspase-8 function, this will induce apoptotic cell death. And caspase-8 has another function. It also can act to prevent RIP3 kinase and RIP1 kinase activation. And this inhibits a necrotic form of cell death, which has recently been termed necroptosis. So if you inactivate caspase-8, say with caspase inhibitors or the endogenous mammalian caspase-8 inhibitor C-flip, then you no longer get inhibition of RIP1 and RIP3 signaling. And this promotes activation of RIP1 and RIP3 kinase activity, and they can autophosphorylate as well as cross-phosphorylate. And it's not fully understood, but somehow downstream of RIP3 and perhaps the generation of mitochondrial-derived reactive oxygen species, this drives a caspase-independent cell death mechanism, which is being called necroptosis, largely because the chemical compound which targets RIP1 kinase activity, necrostatin-1, can block necroptosis from occurring. So it's not really well understood yet how IAPs prevent the formation of this ripoptosome complex, but there has been some evidence from Pascal Myers' lab that they do target RIP1 kinase as well as other ripoptosome components through their E3 ligase ubiquitination activity. So starting off in Jörg's lab, his lab was focused on innate immune signaling and pattern recognition receptor signaling. So I was asking the general question whether these IAP proteins could also play a key role in innate immune signaling. And there were a number of groups asking this question at the time because a lot of the components between TNF receptor superfamily signaling members and pattern recognition receptors, such as toll-like receptors and viral double-stranded RNA receptors, such as RIG-I, a lot of those signaling components are conserved, including TRAFs. So some more recent studies have actually shown a key role for IAPs in pattern recognition receptor signaling. So we have toll-like receptors such as TLR3 and 4 or TLR2 and 4, and it's been shown IAPs can target these complex components such as RIP1 and RIP3 kinase or TRAF3 for ubiquitination. And this allows the formation, again, of a transcriptional response and the production of cytokines. It's not just TLRs that IAPs regulate. Recently, John's lab, postdoc Uli, showed that IAPs regulate NOD1 and 2 signaling. These receptors respond to bacterial peptidoglycans. And here it's thought that it's mostly XIAP, but CIAP1 and 2 may play a small role in ubiquitinating RIP2 and modifying this with ubiquitin chains. And again, this allows a transcriptional, in this case, antibacterial immune response. And finally, now a couple of years ago, it was shown that IAP targeting of TRAFs on the RIG-IL MABS complex, which is localized to the mitochondria, again through ubiquitination promotes NF-kappa B or transcriptional activation of antiviral genes. So these studies were all coming along while I was in Jörg's lab, and I was fortunate enough to be studying another pattern recognition receptor complex, which Jörg and Fabio Martignon, a student in his lab at the time, had discovered. And they called this complex the inflammasome. And they demonstrated this complex regulates the activation of caspase-1 and inflammatory caspase. And two of the key substrates for caspase-1 are precursor interleukin-1 beta and interleukin-18. So what they demonstrated, you have nod-like receptor family members. When these get activated through various bacterial pathogen stimuli, through their purine domain, they can complex this adaptor protein termed ASC. And this ASC protein has a card domain, or a caspase recruitment domain. And this allows a homotypic card-card interaction with caspase-1, resulting in proximity-induced cleavage of caspase-1 and generation of the P20 and P10 active subunits. And this subsequently cleaves the precursor IL-1 and IL-18 into their mature active forms, which are then secreted from the cell. So I should note here the precursor IL-1 and IL-18 is held within the cell and is inactive. So there's not just one inflammasome complex, as I said, there's a number of nod-like receptor family members now that have been demonstrated to form inflammasome complexes, resulting in caspase-1 activation. And some of these aren't all of them, but some of them are listed up here. So we have the NLRC4 inflammasome, also known as IPAF, which can detect and respond to bacterial flagellin to initiate caspase-1 activation, as well as NLRP1B, which can respond to anthrax lethal toxin. And some of these inflammasomes, the ligand actually isn't known yet. So for NLRP6, it's been shown to form an inflammasome to activate caspase-1 and generate IL-1 and IL-18, but it's not yet known what the bacterial or viral ligand is. So one of the most studied inflammasomes is NLRP3, shown here. This is widely studied, and it's been shown to be mutated in a number of autoinflammatory diseases, which are successfully treated with anti-IL-1 therapy. So some of the stimuli for NLRP3 includes the pathogens such as bacterial pore-forming toxins, viral RNA and DNA, as well as yeast beta-glucans. And over the past 4 or 5 years, NLRP3 signaling has really been associated with some more common human diseases or autoinflammatory diseases. So Jörg's lab played a key role in this, and they showed that uric acid crystals responsible for gout activate NLRP3 and to generate bioactive IL-1 beta. And now clinical trials have shown that anti-IL-1 therapy is very successful in treating gout flares. More recently, some of the work which Seth Masters contributed to showed that islet amyloid polypeptide and saturated fatty acids, which are implicated in type 2 diabetes, also activate the NLRP3 inflammasome to generate IL-1 beta. And there's just a number of other common diseases listed here that it's now suspected, although not proven, that NLRP3 signaling and IL-1 may play a key role in their pathogenesis. So the obvious question is, given the chemical and structural diversity of all these molecules that have been shown to activate this NLRP3 inflammasome, is how is NLRP3 sensing all these diverse molecules? So there's actually no conclusive evidence for how NLRP3 is activated. It's fairly clear, I think, based on the data, that all these molecules don't directly bind to NLRP3, and it's probably a more general sensor of cellular metabolic stresses. And what these are really still speculative, but there has been evidence put forward to show that for particulate stimuli such as asbestos and silica, which are phagocytosed through macrophages, these result in lysosomal rupture, and there's protease releases which somehow can activate NLRP3, perhaps through reactive oxygen species generation. For other soluble stimuli, or DAMPs or PAMPs, damage-associated molecular patterns, or pathogen ones, Jürg's Chop published about 2 years ago that this results in altered mitochondrial metabolism and the generation of mitochondrial ROS. And a recent paper has put forward the hypothesis that oxidized mitochondrial DNA is released from damaged mitochondria, and that can bind to activate NLRP3. And this all results in the activation of R1 beta. So as I mentioned, I wanted to study whether IAPs could actually play a role in this inflammasome complex. So we've used here is our Smac mimetic compound, which is called compound A, which we are given by tetralogic pharmaceuticals. And I just want to note here, this antagonizes to some degree CIP1, CIP2, and XIAP. So it's targeting all three IAPs. So for most of these stimuli, they've been done in bone marrow-derived macrophages or dendritic cells. And for most of the experiments I present, I'm looking at a time course of between 5 and 6 hours of stimulation. And for all these experiments, as I mentioned earlier, you require a priming or NF-kappa B signal to drive production of precursor IL-1 beta. And as you can see here, in a normal breasting, this is a bone marrow-derived macrophage, non-stimulated, you can't really detect any precursor IL-1 beta. And you can see here with LPS, or what we call LPS priming, to generate this precursor IL-1, you get a nice induction of precursor IL-1 induction. You then come in with a second stimulus to activate the inflammasome. And here I've used alum-adjuvant, which is a known NLRP3 inflammasome activator. And if you focus here on this Western blot, you can see this is looking at the release of cleaved mature IL-1 beta into the cell supernatant using the known NLRP3 activator alum. And this coincides with the cleavage and release of the active P20 subunit of caspase 1. So you can see here I've done a dose titration of Smac mimetic from 100 nanomolar to 500 nanomolar. And you can see a dose-dependent increase in the secretion of the mature IL-1 beta 17 kilodalton fragment. And this also correlates with the dose-dependent increase in release of active caspase 1. So this suggested the caspase 1 activation, that somehow IAP antagonism was activating an inflammasome. So we started screening inflammasome knockout mice to determine which inflammasome IAPs might be repressing. And so here, to cut a long story short, we've examined NLRP3 knockouts, the NLRP3 adaptor protein ASC, as well as caspase 1 knockouts. And down here, we're looking at IL-1 beta secretion into the cell supernatant. And we've primed, all these cells have been primed with LPS, then treated with Smac mimetic in a dose-dependent manner up to 500 nanomolar. So in wild type, this is bone marrow-derived dendritic cells, you can see a dose-dependent increase in IL-1 secretion, as well as in caspase 1 wild type mice. And you can see that in NLRP3 knockout, ASC knockout, and caspase 1 deficient mice, there's a significant decrease in IL-1 secretion, although it's not complete. And to determine whether this is the mature active form, what we do now is Western blots on the cell supernatants to detect the P17 mature bioactive IL-1 beta fragment. So again, you can see with LPS alone, you get no IL-1 cleavage or secretion, but when you add our Smac mimetic compound A, you generate bioactive IL-1 that's secreted into the supernatant, and this correlates with caspase 1 cleavage and secretion. You can see here, though, in the NLRP3 knockouts, you're completely abolishing the Smac mimetic-induced caspase-1 cleavage and secretion, showing that IAP antagonism is definitely activating the NLRP3 inflammasome. However, as you can see from the Eliza data as well as the Western blot, you still get some mature R1 being released into the supernatant, and this is clearly through an inflammasome-independent mechanism. So obviously, given that the Smac mimetics target all three IAPs, we wanted to know, A, whether this was an on-target effect we were examining, and B, which IAP do Smac mimetics actually target to repress inflammasome activation. So here we've taken the IAP knockout mice, so this is XIAP, CIP1, or CIP2 knockout mice, and we've primed them with LPS, and you can see very little IL-1 secretion in the single knockouts with LPS priming alone. And you can see here we've treated with known NLRP3 inflammasome activators, alum, this is uric acid or nigericin, a bacterial pore-forming toxin, and you can see there's no significant difference in IL-1 secretion in any single IAP gene-deficient bone marrow-derived macrophage. Now this was somewhat surprising at the time I was doing these assays and looking in these IAP knockouts, because we knew from a meeting report that Maya Sellers group in Canada were showing the opposite, and that actually putting forward evidence to suggest that cellular IAP, CIP1 and 2 actually required for inflammasome activation. And this is some of their key data, which was published at the end of last year, and this was published just before our paper came out. And here they've taken, this is BERK3, the gene encoding CIP2, so they've taken CIP2 knockout mice, and you can see for these known NLRP3 activators, ATP, uric acid, or alum, that in the CIP2 knockout mice, they're seeing significant decreases in IL-1 secretion. And I'll just say that we've gone on, we've tried to reproduce their data using the same protocols that they have, but we have yet to reproduce either their in vitro data using CIP knockout mice or any of their in vivo data. And I'll hopefully convince you that she's actually wrong with the rest of my data. So here, what we look at now is, so we're taking these single knockouts and starting to look at double and triple knockout mice. And if we look down in the graphs here, in these LPS-only primed cells, what you can start to tease apart some IL-1 secretion. So this is after a 5-hour stimulus and you can see with LPS priming alone in the XIAP knockouts, there is a small but significant increase in IL-1 secretion in these cells. And you can see if you take now XIAP, and this is CIAP2 double knockout cells, macrophages, and you can see this increase in IL-1 secretion is elevated even more. Now, if you look at this in the context of known NLRP3 activators such as alum, this is still quite low. So if you plot these values alongside alum and Smac mimetic treatments, you can see this hardly registers. Now, if you come on to these single XIAP knockout cells and treat them with Smac mimetic, or in fact, if you treat these double knockout cells with Smac mimetic here to remove the remaining IAPs, so you're removing CIP1 in the double knockouts, and in the XIAPs, you're removing both CIP1 and 2. And you can see now that when you remove all three IAPs, you get a massive increase in IL-1 secretion. And this suggests to us that it's really CIP1, 2, and XIAP that is required for maximal IL-1 beta activation. And Kate Lawler, who's joined Davo's labs recently, demonstrated this quite nicely. So here Kate's taken, again, she's taken the double knockout mice, and here instead of treating with Smac mimetic to remove the remaining IP, she's examined triple knockout mice. So these are XIAP, CIP2 double knockout mice, and then CIP1's knocked out with lysine-free, CIP1's floxed. And you can see here now with LPS treatment alone, you get very significant IL-1 secretion in the triple knockout mice relative to the double knockout mice. Now, and again, if you now treat these double knockout mice, shown in green here, with Smac mimetic to remove CIP1, then IL-1 levels go up and somewhat resemble the triple knockout mice. And what the other point of note here, I think, is that this is compound LPS priming with compound treatment alone here, and as well as here. And you can see you're getting much less IL-1 secretion when you treat with compound A, which supposedly targets all three IAPs. So it just suggests that the Smac mimetic targeting of all three IAPs is quite inefficient when you're comparing them to triple knockout, genetic knockout, bone marrow-derived macrophages, which might not be a bad thing in the clinic, in actual fact. And this is just a 24-hour time point showing pretty much the same trend. So, of course, you might wonder whether this is just general cell lysis and release of pro-IL-1, what we're measuring in these ELISAs. But what you can see here is we've run the supernatants from either the wild type, all the single knockout mice, as well as the double knockout mice. This is the XIP, CIP2, or the CIP1, CIP2 double knockouts, or the triple knockouts. And I'll just get you to focus on the point of this Western is showing that with LPS priming alone, just as we just showed you with the Eliza data, with LPS priming alone, you're getting significant cleavage and secretion of the mature 17 kilodalton IL-1 fragment. And this again correlates in these triple knockout mice with a nice induction and secretion of active caspase 1, which you don't see really in any of the other knockouts to any significant extent in comparison. If you look at the cell lysates from these supernatants, now we're looking at the cell lysates, you can see, if you ask, well, are these cells actually dying? And the answer for the triple knockouts, at least, is clearly yes. So if you look at, the triple IAP knockout bone marrow-derived macrophages, you can see significant caspase 3 cleavage with LPS treatment alone, as well as PARP cleavage. So one of the obvious questions that we needed to address was whether the IL-1 secretion and activation actually precedes cell death. And I've done quite a bit of work to convince myself this is the case. And I'm just going to show you one bit of data in that. Here we're looking at propydium iodide uptake and annexin 5 staining to look at cell death. And this is LPS primed followed by Smac mimetic treated cells for 8 hours. And this is at a dose which induces clear IL-1 secretion. And at this time point, these cells are the plasma membrane is not compromised at all in comparison to, say, known cell death inducers such as staurosporine or cyclohexamide. So definitely, although IL-1 activation and cleavage for all inflammasome stimuli go hand in hand with cell death occurring, what's, I think, becoming more accepted now as more papers come out is that the IL-1 cleavage and secretion precedes cell death. These cells, although it precedes cell death, these cells are nevertheless destined to die. So does this occur in vivo? So tetralogic generated data showing they could inject the Smac mimetics and within 7 hours they could detect serum level increases in both IL-1 alpha and IL-1 beta. So this is a dose-dependent effect, and this is looking at IL-1 induction in the serum of Smac mimetic injected mice as well as IL-1 beta induction. And I should really point out all these experiments wouldn't have been possible without Lynn, a recent postdoc in John's lab, and she went to, I think, an enormous effort to generate all these double and triple macrophage deficient mice. So here, this is some of Lynn's work, and what she's done here is she's generated XIP, CIP2 double knockout mice, and then knocked out CIP1 using lysine-Cre, so knocked it out in myeloid cells. And you can see here these triple knockout mice are extremely runted, and they have a loss of hair and a very much pro-inflammatory phenotype. And if you look at IL-1 levels in these mice, in the triple knockouts, you see elevated serum levels of IL-1. And she's also observed elevated IL-1 serum levels in the serum of these double knockout mice, and I think these were derived in non-pathogen-free conditions, so it'll be interesting to see whether this holds true in pathogen-free conditions. So anyway, so this is where we're up to with our model, is that these IAPs repress IL-1 activation, and they do so by acting all together. So you require CIP1, CIP2, and XIP to repress IL activation, and you need to remove all three for inflammasome activity. So somehow it activates the NLRP3 inflammasome to generate bioactive IL-1 and also generates IL-1 through this other mystery protease. So the next question was, what could they be targeting to prevent this IL-1 activation? So as I mentioned before, the IAPs have been shown to repress the spontaneous formation of this ripoptosome complex, which contains the RIP1 kinase, RIP1, and as well as RIP3 kinase, as well as caspase 8. So we asked, can we detect ripoptosome formation in primary cells? Because all the previous work had been done in cancer cell lines, so we're looking at bone marrow-derived macrophages again. And can we see the ripoptosome in bone marrow-derived macrophages when we remove IAP function with Smac mimetics? So we've done this by immunoprecipitating caspase 8 to pull down the complex, and this is just the IP here showing caspase 8 being pulled down in all lanes. And then we've looked at RIP1 association with caspase 8. So what was shown upon ripoptosome formation, you get an increase in the spontaneous complexing of caspase 8 with RIP1. And you can see here, this certainly held true in our bone marrow-derived macrophages. So here, this is without LPS priming, with Smac mimetic treatment for 1.5 hours. And you can see you get a significant increase in RIP1 binding to this immunopurified caspase 8. And again, this increases after 3 hours. And if you follow this complex formation, after LPS priming, complex formation is increased and accelerated. So you get an even more significant increase in RIP1 association with caspase 8. So this suggested to us that the IAP removal in macrophages induces ripoptosome complex formation. So next we tested, because we had available to us the RIP3 kinase knockout mice, whether this was involved in IL-1 secretion. And another real reason for testing the RIP3 knockouts was, as I mentioned at the beginning, RIP3 has been hypothesized to promote the generation of mitochondrial ROS to promote cell death or necroptosis signaling. And Jörg's lab had shown a couple of years ago that mitochondrial generated ROS may also play a role in inflammasome activation. So this is just a Western blot, again, similar to what I showed you before, in that we're looking at a dose-dependent titration of our compound from 100 to 500 nanomolar, and we're looking at secretion of the 17 kilodalton I1 fragment into the supernatant. And this is in wild type, this is in dendritic cells, you see I1 cleavage and secretion. And this also, again, you see this correlates with caspase 1 cleavage and secretion. And you can see that this is largely deficient in RIP3 knockout-derived cells. At these higher doses and with increased cell numbers, you still get some IL-1 and caspase 1 cleavage and secretion, but it is very much diminished upon deletion of RIP3. So as I said, the RIP3 has been implicated in the generation of mitochondrial ROS. So we next asked whether given the implication of the hypothesis that mitochondrial ROS may play a role in IL-1 activation, we've treated here bone marrow-derived macrophages with a number of antioxidants. I won't go through them in detail, but these are 4 different chemical antioxidant compounds. And again, we're looking at IL-1 cleavage and secretion to the supernatant. So you can see with our control lane, Smac mimetic induces IL-1 cleavage and secretion, as well as caspase 1 cleavage and secretion. And you can see when we treat with these 4 different antioxidants, this largely prevents both IL-1 secretion as well as caspase 1 secretion. This suggested to us also it's not only blocking caspase 1 or the NLRP3 inflammasome, but it seems to be blocking the caspase 1 independent IL-1 secretion. So to show whether this is the case, what we've done here is taking our NLRP3 inflammasome knockout macrophages and done a dose titration of increasing antioxidant. And you can see here this caspase 1 independent IL-1 secretion, as measured by Eliza, again is decreased by the titration in of antioxidant compound, suggesting that ROS is involved in both the NLRP3 inflammasome activity as well as NLRP3 inflammasome independent IL-1 activation. And to determine whether this was RIP3 kinase mediated generation of ROS, which other people have proposed, we've measured ROS using a fluorescent probe in bone marrow-derived macrophages. And you can see with LPS priming and compound A or Smac mimetic treatment, you see a pretty robust increase in ROS generation in wild-type macrophages. And this is largely prevented in RIP3 knockout macrophages. So this is some preliminary data which we're following up on at the moment. So this is looking at two recent papers came out, one by Zadal Wang's group in Cell, demonstrating that downstream of RIP3 kinase, they've proposed that mixed lineage kinase domain-like protein acts downstream of RIP3 in executing the necroptotic signaling cell death. And we were fortunate enough that Warren Alexander's groups generated these MLKL knockout mice. What's really surprised me, and initially I wasn't so excited to do these experiments, but Kate really drove ahead and said we should do them anyway, because I thought they'd just be the same as the RIP3 knockouts, but it turns out they're not. So here we've got wild-type macrophages, RIP3 knockouts, MLK knockouts, and TNF knockouts in parallel, and examined them again with a dose-dependent increase of Smac mimetic. And you can see in wild types, this is increase in IL-1 secretion is largely prevented in RIP3 knockouts. It still occurs in TNF knockouts, so it's a TNF independent mechanism. But you can see in the MLKL knockouts, they really seem in this at least one preliminary data set respond very similar to wild-type macrophages. So this suggests, at least in the terms of inflammasome activation and IL-1 signaling, that maybe there's bifurcation of RIP3 and MLKL signaling. And this is something we're pursuing at the moment. Nevertheless, there was still the remaining question of what is this alternate protease that might be cleaving and activating IL-1? As you can see, this is just what I showed earlier, that caspase-1 knockouts still generate significant amounts of IL-1 when you remove IAPs. And this Jörg, at the time when I was showing Jörg this data, he was quite enthusiastic in pursuing this, because I think what's becoming more apparent recently is that a lot of these NLRP3 activators, although they're required for IL-1 activation in vitro, that when it comes to mouse models of disease such as gout and arthritis, that they actually occur independent of caspase-1, although they still require very much IL-1 signaling. So it suggests in vivo that caspase-1 might not be so dominant in generating active IL-1 as previously thought. So we followed this up. Now, what we observed was that ZVAD, which is a pan-caspase inhibitor, so it pretty much blocks all the caspases in the cells. And when we treat macrophages with ZVAD, this completely blocks the IL-1 cleavage and secretion in wild-type macrophages. So you're comparing, these are just two different Smac mimetic compounds, and you can see nice cleavage and secretion in untreated cells, but when you pre-treat with ZVAD, you're blocking this secretion. And this is just another antioxidant, so you can ignore these lanes. And this also, as expected, will block the IL-1 cleavage and secretion. And more importantly, if we now take caspase-1 knockout macrophages and treat these with Smac mimetic, again, we can see in the absence of caspase-1, you still get significant IL-1 cleavage and secretion. And ZVAD, again, will block this in caspase-1 knockouts, suggesting that another caspase can actually cleave and generate bioactive IL-1. So what we ended up doing, because the caspase-8 knockout mice are lethal and it appears that caspase-8 is actually required for macrophage differentiation. So we've taken an immortalized macrophage cell line, and this cell line lacks ASC, so it can't activate NLRP3 anymore and can't therefore activate caspase-1 with Smac mimetic treatment. And you can see this here with our Smac mimetic treatment, you get significant IL-1 cleavage and secretion in the absence of caspase-1 cleavage and secretion. But what you can see after a number of Western blots and looking at various other caspase gene deficient mice is that we always observe very nice cleavage of caspase-8. And this is a cleavage specific antibody, so it only detects the cleaved caspase-8 product. And we were able to demonstrate in vitro, at least, that recombinant caspase-8 could actually cleave to generate bioactive IL-1. This is in contrast to other recombinant caspases, such as caspases 3 and 9. So what we've done now is knocked down caspase-8 using a lentiviral shRNA constructs, and we've got 5 different shRNA targeting constructs here, and we've treated, this is our scrambled or TNF shRNAs, and again these cells respond by secreting mature R1 in response to Smac mimetic treatment in the absence of caspase 1 cleavage. And you can see here we now knock down caspase 8. Just about 4 out of 5 of these constructs reduced the amount of cleaved and secreted R1. And there was really only one construct in this cell line was quite sick looking and constitutively secreted R1. So at least 4 out of 5 shRNAs, which all knocked down caspase 8, prevented R1 secretion in this caspase 1 null cell line. So this suggests to us at least that you have a caspase 1 dependent R1 cleavage as well as a caspase 8 dependent R1 cleavage. Got a few more minutes. So if we look in these cells in parallel, you might have noticed before that the RIP3 knockouts produce less R1 compared to caspase 1 knockouts. And this implies really that not only can caspase 8 negatively regulate RIP kinases such as RIP1 and RIP3, but perhaps RIP3 activity is actually also required for optimal caspase 8 activation when we treat macrophages with Smac mimetic compound. And this was a bit of, I think, heretical idea, but there was actually some evidence for this occurring. And these are the initial 2 papers identifying RIP3 kinase, and both of these showed that overexpression of RIP3 actually induced apoptosis. And both of these papers demonstrated, one from Vishwadixit, demonstrated that RIP3 may associate with caspase 8 and in fact can result, its overexpression at least can induce caspase activation. And recently Donya in Davo's lab has actually to some degree reproduced this data, but not only that, but extended it significantly. So I'll just show a couple of slides of her data, what she's done. So she's taken wild-type RIP1 knocking fibroblasts, murine embryonic fibroblasts, caspase 8 knockout fibroblasts, or RIP1 knockout fibroblasts, and she's reintroduced into these cells a tamoxifen-inducible flag RIP3 construct. So you can see here she's induced with tamoxifen and she gets nice induction of RIP3 in wild types, caspase 8s and RIP1 knockout fibroblast cell lines. Now you can see with induction of RIP3 alone she doesn't see any significant PARP cleavage or caspase cleavage. But you can see if she just adds TNF on top of these cells that are expressing RIP3, adds TNF on top of them, you can now see significant PARP cleavage as well as caspase 3 cleavage. And this is obviously increased if she now comes in and titrates in Smac mimetic compound. What she showed now that this is a caspase-dependent PARP and caspase 3 cleavage, because in the caspase 8 knockouts, when she induces RIP3, here adds in TNF or combines it with Smac mimetic, she now gets absolutely no PARP cleavage and no caspase 3 cleavage. And I don't want to, and pretty much this can occur independent of RIP1. So TNF somehow signals to RIP3, which can result in caspase activation. You might say, well, this is just through TNF and FAD and caspase 8 activity, but what Donya and Wendy and Davo have now gone on to further demonstrate that if you remove TNF from the equation, so here they've made a construct which is a dimerizable RIP3 construct. So you add in the antibiotic kumamycin, this induces RIP3 dimerization and activation. So you can bypass the requirement for TNF. So here, if you induce this construct by itself, you get some PARP cleavage, so this is in the absence of TNF, as well as caspase 3 cleavage. Now, if you dimerize this RIP3 construct, you get increased PARP cleavage and increased caspase cleavage, which really suggests that at least RIP3 activity can promote caspase activation. And we wanted to know with endogenous RIP3, at least when we activate RIP3 endogenously, can this promote caspase 8 activity as well, which will then generate bioactive IL-1. And this paper gave us a clue. This is from Avi Ashkenazi's group in Genentech, and he demonstrated that this Cullen E3 ligases can ubiquitinate caspase 8, and this ubiquitinate ubiquitolated caspase 8 segregates into lipid raft membranes, and that this ubiquitination and segregation is required for optimal caspase 8 activation. And we were quite lucky in that our cleaved caspase 8 antibody worked extremely well and could pick up this smearing of caspase 8, which we think is the ubiquitolated caspase 8. And initially we were a bit perturbed, because if you just have a look in these lanes here, this is wild type cells treated with Smac mimetic looking at cleaved caspase 8. You get nice caspase 8 cleavage to P43 and P26 kilodalton fragments. And if you compare that to the RIP3 knockouts, it's fairly equivalent. We sometimes see a little bit of a decrease in caspase 8 cleavage, but you would say that this isn't that significant. But what we did notice a big difference in is if we now look at this. So this is compound A treatment with 200 nanomolar or 400 nanomolar. And again, you see significant caspase 8 cleavage and what we presume is this smearing or ubiquitination of caspase 8. And at 400 nanomolar, this is increased. And if you compare this directly to the RIP3 knockouts, this is in total cell lysates. So if we're comparing this lane and this lane, you always see significantly decreased caspase 8 modification. And again, this is at 400 nanomolar. You get nice caspase 8 modification, which is decreased in the RIP3 knockouts. And what further suggested that to us that this might be the active caspase 8 was that, consistent with Avi Ashkenazi's group's data, this cleaved and modified caspase 8 fractionated preferentially into Trident X100 insoluble membrane domains. And finally using a fluorogenic-based caspase 8 substrate assay, we could see that over time, when we prime macrophages with LPS and treat them with compound A afterwards, the RIP3 knockouts consistently show decreased caspase 8 activity in this fluorogenic-based substrate assay. And this was observed a number of times. And it wasn't a complete blocking caspase 8 activity consistent with only a decrease in modification, but it certainly was significant. So finally, I'll present some in vivo, because you might ask what's the relevance of all this physiologically or in some disease models. So I'll present some data that Kate Lawler's recently generated in Davo's lab to suggest that this mechanism might actually play a role in an acute arthritis model. So this is a KBYN-induced acute arthritis, and this arthritis model has been well documented. This occurs completely independent of caspase 1. So you induce this arthritis in caspase 1 knockout mice, and they develop arthritis similar to wild-type mice. Yet this model is largely dependent on IL-1 signaling. So again, if you take IL-1 knockouts, they show reduced arthritis compared to wild-type mice. So it's clearly a caspase 1 independent mechanism for generating active IL-1, which is largely responsible for driving this disease. So this is Kate Lawler's preliminary data. So she's taken 9 mice of each genotype, injected them with KBYN serum, and looked at the development of arthritis over time. And you can see there's clearly a decrease in arthritis development in the RIP3 knockouts. And she's measured this in an independent fashion by looking at myeloperoxidase activity. So these mice are injected with luminol, and then the bioluminescence is read off in vivo and quantified. And again, this is a marker of immune cell infiltration into the joints. You can see this is significantly decreased at these time points in the RIP3 knockouts. And this is just represented here in an image of the wild type and RIP3 knockout mice with the intensity of myeloperoxidase activity in the joints. And again, Kate went on and dissected the ankle joints and looked at IL-1 secretion from these ankle joints over time. And you can see there's IL-1 secretion in wild-type mice, and in the RIP3 knockouts, this was significantly reduced. And this appears to be in contrast to TNF secretion in these arthritic ankle joints, where TNF levels weren't really reduced. So this is some work that we're hoping to follow up on in the future. So I'll just finally present our final model it's still a working model, and there's a lot of question marks still, especially regarding the role of MLKL in this pathway now. So we think that when you antagonize XIP, you get a little bit of IL-1 activation, but really what I've shown you is that you need to remove all three. And this results in what we think is a ripoptosome or ripoptosome-like complex. And then RIP3 drives ROS production, and this can promote the activation of the NLRP3 caspase-1 inflammasome to generate bioactive IL-1. But this complex also promotes caspase-8 activity, and I hope I've showed you data to help convince you that RIP3 may also modify and promote caspase-8 activation. And this can independently result in bioactive IL-1 production. So what I think, this complex is not just a cell death signaling complex which will execute apoptosis or necroptosis, but I think when it's formed in vivo, it may play a key role in initiating IL-1 dependent inflammation. So I'd finally like to acknowledge all the people, and really this work couldn't have been completed without a large amount of help from various groups. This, as I mentioned, was largely initiated in Jörg Chopp's lab, where I was for almost 2 years. And these people in Jörg's lab, especially Greta and Olaf and Angie, helped generate a lot of the data. Back at WeHi, this wouldn't have been possible without the IAP gene knockout mice generated by Davo and John. And as I mentioned, Kate Lawler has actually generated a lot of data now following up from this work, which is going to be interesting to follow up on. Also, Lorraine O'Reilly, some of the data I haven't shown, but her and Kylie Mason helped provide back-to-back double knockouts to examine. I'd like to acknowledge Tetralogic for the Smac medic, and also Ian Gentle, who's part of Jörg Hacker's group in Germany, who helped with looking at caspase-8 cleavage of IL-1. So, thank you.
We've got time for a couple of quick questions. I might kick it off. Maya Sally, whose group you contradict, you say you can't see the same things as she sees. She proposes that it may be something to do with a late effect of loss of IAPs versus an early effect, and she says that when she loses IAPs early, you don't activate the inflammasome, but if you lose IAPs for a little bit longer, then you do activate the inflammasome. Do you have any data to actually deal with that issue? And I should point out that in her supplementary figure, she actually sees very similar data to you.
That was with the BB6, I think.
Yeah.
So this is a Genentech-derived Smac medic. So we have tried to exactly reproduce her protocol, as you're aware of, and we follow her time points, which include very early time points, using the same inflammasome stimuli as she does in our IAP deficient mice, and we never see a significant decrease in IL-1 secretion, which is what she reported. I haven't gone into it all. Obviously, part of her model also, because IAPs bind TRAF2, she also proposed that TRAF2 is in part of that caspase-1 complex, and TRAF2 is also required for optimal caspase-1 activity. And we've subsequently also analysed TRAF2 knockout mice, and again, we can't see any defect in IL-1 activation. And we've gone further than that. Kate Law has actually done some studies injecting uric acid crystals into mice to look at IL-1 induction during MSU-induced inflammation and neutrophil recruitment, and again, she sees no significant difference, at least in the one experiment, between wild type and IAP knockout, single CIP1 knockout mice. So, even the early and late data time difference that she was reporting, we never yet observed. So
in summary,
it could be, you
cannot reconcile her data.
The only reconciliation I could offer was that the IAP knockout mice she's using were derived on a different genetic background and were then back-crossed. So that's the only explanation I could think of, is that there's some background differences between her IAP knockout and our IAP knockout mice.
So it's caspase-8, pro-caspase-8 has death effector domains, and it's known that FAD, which has death-affected domains, can activate the caspase 8. RIP has been proposed to activate caspase 8 by binding via its death domains to the death domains of FAD, which then activates caspase 8. How does RIP3, which doesn't have death domains or death-affected domains, activate caspase 8, and how does it do it in a RIP1 knockout?
It could be by binding FAD. People have reported RIP3 can complex FAD, but
not via a death domain, correct?
No, I don't think they've defined which part of RIP3 could interact with FAD. There's also data which I know Donya has generated, which suggests RIP3 could bind TRAF2. And again, it's unclear how that would occur, but that's two possible scenarios.
And then how would TRAF2 activate caspase 8?
Well, that would link TRAF2 to TNFR1, which could then... So TRAF2 can bind TNFR1, which would bring in RIP3 and initiate. But your guess is as good as mine.
Another take here. Obviously, the Smac mimetics are in phase 2 clinical trials. So, whereas you showed data from Tetralogic that in mice, you saw induction of these inflammatory cytokines. Clearly, in humans, this inflammatory response is either manageable or less. Do you think that it's less in humans, or that it's just a question of manageable?
I think, I mean, the data we have now with other Smac mimetics that other companies have developed suggests that, as I showed, even in vitro assays, they're nowhere near as efficient, at least at inducing IL-1 production as knocking out all 3 IAPs. So it really suggests that even in vitro, what you'd suspect is going to be higher doses than in vivo, that their efficacy at antagonizing IAPs resulting in cytokine production is going to be far less than what you can ever observe using a genetic knockout model. And the question still remains how much, I think, is going to occur, how much inflammation will occur in people, and to what extent. And there's always, it could be harmful, but there's also the possibility if you get a inflammatory type macrophage within the tumour microenvironment producing lots of TNF and IL-1, which can also drive TNF, that will actually promote TNF-dependent cell death of tumours, cells which have lost their IAPs due to the Smac treatment. So I think it's definitely still open.
So it could be a positive.
It could, you could always say speculate.
I don't mind if you've got a good question. Everyone else sit for one more minute. Dave.
How much of the arthritis data do you think is due to a difference in IL-1 versus a response to TNF, given the published data in immunity on responses in RIP3 knockout?
Well, we want to address that by making RIP3 TNF double knockouts and doing the modelling. Because, as you said, TNF definitely goes up and it can contribute to that model. So the plan is the RIP3 TNF knockouts should be completely viable, and we should be able to address that. And hopefully within the next year.
But your data showed that in the RIP3 knockout, the TNF
was normal.
It didn't change.
So we would like to rule out the contribution of TNF to that disease phenotype.
And even bring it down further.
But it wouldn't need to change. You would have similar levels of TNF, but you would have the downstream effect.
Yep.
Okay, well, thank you very much for your attention. Thank you.